Welcome to our lecture on puberty. The next slide will show the hormonal changes in the male, particularly in the stallion. This diagram shows the changes in the hormones as the stallion approaches puberty. So this is age in weeks. So it is said that the stallion will reach puberty at about nearing 80 weeks of age. So as you can see in the diagram, there are changes in the level of FSH and LH prior to the onset of puberty. So as shown in the diagram, even before the stallion reaches puberty, it has already an increased level of FSH and LH. We also have here, as shown in this stallion, we have an increased gonadotropin prior to the onset of puberty. We also have here, prior to the onset of puberty, there is a decreased level, minimal level of testosterone that is found in the blood. And as the animal approaches puberty and until it reaches puberty, there is a gradual increase in the level of testosterone, as shown in the diagram. Prior to puberty, there is already a minimal amount of um, testosterone present, and this is important for the Sertoli cells to mature and multiply, and for spermatogonia to begin to divide. So testosterone at this point of time is considered to be low. As the animal approaches puberty, so the this testosterone will increase now, and of course that is brought about by the LH and the FSH that are important for the increase in the level of this particular hormone. So as the Sertoli cells and the Leydig cells mature, then the FSH increases now at this point. Testosterone will also increase. This is the green line. And we also have the LH. LH is first down and then increases after that. At puberty, the sperm begins to be seen in the ejaculate at this point. And there is, of course, a large increase in testosterone during puberty. Genetics is also a factor for the onset of puberty. One way to look into genetics is to observe the breeds within the species. The breed has an important influence on the age at which puberty is attained in both the male and the female. So as shown in the diagram, you have here the different breeds of cattle, swine, and sheep. We also have here the average age at puberty in months for female animals and male animals. So as you can see here, generally, the male animal reaches puberty at an earlier age compared to the female animal. And there is a wide range in the onset of puberty for the female and the male animals between of the same species. This is also true for swine. So genetics can influence the age at puberty. Another important factor that determines puberty is the nutritional status of the animal. So adequate growth in replacement heifer is necessary for reaching the pubertal status prior to the breeding period. The body weight is very important. It is a primary determinant of puberty attainment in heifers. Beef heifers usually achieve puberty when they reach 55 to 60% of the mature body weight. At this point, there should also be the animal should also begin fat deposition and preparation for the puberty and eventual conception and parturition. This diagram shows the effect of glucose on the hypothalamic control of the GnRH secretion, particularly the LH pulse. So in this experiment was conducted in ewe lambs, and they utilized 2-deoxyglucose as the main treatment to determine its effect on the LH pulse. So 2-deoxyglucose is an analog of glucose that is not metabolized, meaning that it will not enter the glycolysis. It will not produce pyruvate and ATP, and it will not produce energy to the animal. So the animal will have no a sense of starvation during when the 2-deoxyglucose will be administered to that particular animal. So before 2-deoxyglucose, the LH amplitude and the frequency is normal, but during the 2-deoxyglucose, because this substance competes with glucose for glycolysis, so there again the LH will be reduced significantly. When the animal will be administered or will be given GnRH at the ninth hour of the experiment, as you can see here, there is a significant increase in the LH pulse because the GnRH will have a positive effect, will stimulate the production of the LH. So going back to the diagram, so at this during the administration of 2-deoxyglucose, there is a decrease in the amplitude and the frequency of the LH pulse. So note that giving GnRH at this point will increase the LH production, and this is due to the effect of nutrition on LH and GnRH pulses. This diagram shows the metabolic signals that influence the GnRH neurons. The GnRH is important for the production of the hormone GnRH, and these are the signals that are important for the GnRH neurons. We have the glucose, the leptin. Of course, the leptin comes from fat and the blood fatty acids. So this is an illustration of the metabolic signals influencing GnRH neurons at the prepubertal or the starvation phase. So the glucose, leptin, and the blood fatty acids are also regarded as the metabolic signals, and they are converted to neural signals in the hypothalamus. Of course, we have the important neurons, neuropeptide Y for leptin, glucose sensing neurons for blood glucose, and the fatty acid sensing neurons for the blood fatty acids. So these neurons will synapse with kisspeptin neuron. Kisspeptin neuron is a neuropeptide, or kisspeptin neuron is of course a neuron that produces kisspeptin. Kisspeptin is a neuropeptide expressed in these neurons, and these are important for control of the GnRH secretion and play a role in the regulation of the hypothalamic pituitary gonadal axis. So there are three presynaptic neurons that stimulate the GnRH neurons. We have the leptin-sensitive neurons, and these are the neuropeptide Y. We have the glucose-sensitive neurons and the FA-sensitive neurons. At this point, during the prepubertal or the starvation phase, there is not enough blood glucose, leptin, and blood fatty acids, so there is not enough signal that is sent to the neurons, and this will be converted to neural signal. So there is not enough GnRH, or there is a low level of GnRH that is produced at this time. So at this point, no, these signals has a negative feedback to the production of the GnRH. In the prepubertal or the starvation period, there is a low level of blood glucose, low fats, leptin, and low fatty acids. All this will have a negative effect to the GnRH production. That is why before the onset of puberty, or for puberty to be attained, there must be a sufficient level of these metabolic signals in the body. At the post-pubertal period, there is high level of blood glucose, leptin, and blood fatty acids, and all of these will have positive feedback to kisspeptin neuron and to the GnRH neuron to produce more GnRH. For the question 4, metabolic sensing neurons control GnRH release by synapsing with which neurons? The kisspeptin neurons will be the one that will synapse with the different type of neurons for glucose, leptin, as well as fatty acids. This diagram shows the relationship between the plane of nutrition growth ADG with the onset of puberty in dairy heifers. The black curve, as shown in the diagram, represents a high plane of nutrition. At the high plane of nutrition, the animal gains about 2 pounds per day ADG for 6 to 8 months uh, and attain puberty at 6 to 8 months. At the red curve, that represents a moderate plane of nutrition. So at this type of nutrition, the heifer reaches about 1.5 pounds per day ADG, and it will attain puberty at a later age of 9 to 11 months. For the blue curve, this represents a low plane of nutrition with a 1.2 pound per day average daily gain. And this is brought about by restricted feeding or low quality feed. And most of these heifers will reach puberty at an even later stage of 12 months, but they will be also too small for successful pregnancy and parturition, although they are capable of becoming pregnant. Animals reach puberty at different body weights. All female mammals must acquire a certain body size before the onset of puberty can be initiated. One factor that impacts the development of the hypothalamic neurons, GnRH neuron, in the female animal is the development of the threshold body size and/or composition. For dairy cattle must attain a mature body weight of 30 to 40%. So it must attain 30 to 40% of its mature body weight in order to reach puberty. 55 to 65% must be attained by the beef cattle in order to attain puberty, and for sheep it must attain 40 to 63% of its mature body weight in order to uh, reach puberty. The environment and the social conditions are also very important for the onset of puberty. For the environment and the social conditions, we have two factors. We have number one is the number of females housed together, and we also have the exposure to a male animal. This diagram shows the effect of large groups versus small groups on the onset of puberty. On the left, we have the large group of about 10 gilts. On the right, we have small groups of 3 gilts. When we are going to interpret the result, so the gilts housed in larger groups of more than 10 will reach puberty at 28 weeks, which is the recommended age, 28 or 7 weeks. When we are going to reduce the number, or when you are going to house them in small groups of 3, uh, the gilts will attain puberty at a later age, that is an additional 4 weeks or a month, and that will be 32 weeks. We also have the effect of the male. So gills that are housed in either small or larger groups and exposed to a boar will enter puberty at an earlier age than their counterparts that are not exposed to a boar. So in this diagram, so we have here the large group housed with a boar in fence, so that will reach puberty at 24 weeks. Then a small group with a boar, then it will also reach puberty at about 24 weeks. An important point to recognize is that the presence of the male, either in visual contact with the females or in direct physical contact with them, will hasten the onset of puberty in gilts. Nebraska researchers have shown that bulls accelerate the onset of puberty in beef heifers. So this experiment was conducted to determine the influence of growth rate and bull exposure on age of puberty. So the one on a red part here represents the moderate growth rate of the animal, and the blue one is a high growth rate of the animal. And this y-axis represents the age wherein the animal was able to reach puberty. So the shorter the bar graph, the earlier the onset of puberty in that particular animal. So this portion here is heifers only. There is no boar. There is no bull rather. While in here we have bull exposure. So there is an interaction between the growth rate and exposure to the bull as shown in the figure. For example, heifers with a high, no, this portion one here is a high growth rate and exposure to the bull for 6 months reached puberty at about 375 days, so shown in this diagram, 375 days. Those with moderate growth rate with bull exposure. So moderate growth rate with bull exposure reached puberty at about 422 days. Heifers with a high growth rate that were exposed to the bull attained puberty 74 days earlier than heifers with moderate growth rate and no exposure to the bull. So the shorter the bar graph, it means that the animal has a high growth rate and it has a bull exposure. So it attained an earlier age to puberty as compared to those heifers that has a high plane of nutrition, high growth rate, but no bull exposure. So therefore, it indicates that the presence of the bull hastens the onset of puberty in the beef heifer. Social cues significantly impact the onset of puberty in many mammalian species. This is caused by olfactory recognition of pheromonal substances that is present in the urine. The presence of certain pheromones produced by the same or opposite sex alters the onset of puberty. The pheromones will send its message to the hypothalamus via the olfactory and the vomeronasal organs. These are the recommended age to breed different animal species. For the heifer, that is 15 months or 65% of the body weight, mature body weight. We also have the bull, 15 natural mating, 12 months for AI. So when the bull is for AI, you can use that at the age of 12 months. That would be more later when we are going to use the bull for natural mating. For filly, the recommended age to breed is 2 to 3 years. Of course, this will depend on the breed of the animal in all species. For the gilt, that is about 8 to 9 months. For the boar, that is 9 months. Can exposure to a male decrease the age of puberty in a female pig, cow, or human?